Can we proceed?
Yes, you can start, sir.
Okay. Now, I'm going to discuss with you the immunity to some of the pathogens. Um, and I'm hoping you've done some bit of microbiology so that we can be able to walk through the talk with a bit of knowledge. Now, in case you've not done microbiology, there are a number of concepts and definitions which you require for you to understand about how our immune system, how bacteria, how we gain immunity against some of the pathogens. One of the concept of definition you need to understand is what's a disease. And a disease is actually the results of a damage in presence of a microorganisms or their product. So you can get disease because of either a pathogen, presence of a pathogen, or their products. And many of them are going to be within that spectrum. So then there is infection. And infection is merely the presence of a pathogen without causing the disease. And a good example is when COVID came up, you heard that there were people who actually could get COVID infections without necessarily having a disease. And there are those who got COVID infections, ended up including dying. So those concepts requires to be understood very well. Then you can get a stage where actually a pathogen merely actually colonize you. It coexists with the host without causing an infection or causing a disease. And sometimes you can enter in a carrier state. You are able to get colonized, transmit the pathogen to somebody else, but you do not have an apparent infection. Now, the final one I would want us to run through is what's a pathogen. Is technically is any pathogen with a potential to cause a disease. And using that definition, then when you go to microbiology, you understand there are actually pathogens which exist and do not cause a disease, but later on they can change and cause a disease. So those are important concepts for you to understand. Then you need to go on and understand what are the sites which are commonly colonized. One of them is GIT system. Then there is upper respiratory tract, then genital tract, and the skin. Those are actually commonly colonized. There are those areas which stay sterile, including blood. Blood should actually be sterile. Then cerebrospinal fluid, the interstitial fluid and spaces, and lymph. And when you end up getting any pathogen in there, more often than not, you'll find that you'll be so sick because those sites are supposed to be sterile without anything. And you understand why. Now, when you do that, for you to understand what will happen in each of the stage of a disease, you need to then characterize different stages of the pathogen. And these are well known. One of them, any pathogen has to encounter the host. So that relationship requires to be built. And when you encounter the host, then there is the entry of the pathogen to the host. It may spread or not spread. It may stay within localized place of entry or spread. And it requires to multiply, because if you don't multiply, then you're going to have issues of how the few pathogens which have entered are going to evade the immune system. Then obviously is the evasion of the immune system. The number of them will actually cause damage, and it is always suitable if a pathogen can either move to a new host or keep moving within the same host. So those stages of a pathogen, for them to cause disease are important. And in each of those stages, the immune system has specific methods which it uses to prevent the encounter, to prevent the entry, to prevent the spread, to prevent the multiplication and damage. So there are those specific areas which you need to remember, and immune system has put up procedures to actually prevent damage. Sorry, cursor is refusing to move. Now, let's discuss each of them, and then I will tell you how the immune system does avoid each of these. Now, you can, there are two ways of encounter. It can be from outside there or from inside. So the terminology of exogenous and endogenous infection and the concept of normal flora becomes very critical. If a pathogen comes from outside, we call that exogenous. If it's within you, it's called endogenous. And there are different sources of where the pathogen can come from. And these ones you know of very well. Now, you know food, you can either get a pathogen from eating food, contaminated water, you can breathe it, and you can get it if you get into contact with body fluid like HIV. You could get a pathogen from insect bites or from animals or fomites. And you need to understand that their likelihood of having a reservoir within the environment. So where did it come from? And one of the concepts of even COVID is trying to understand where actually COVID originated from, which animal transmitted or passed the COVID virus from the bats to another animal to the host. Then when you understand that, then humankind can put measures in place to eliminate the sources of the pathogen. And for example, it's mosquitoes, which are carriers of malaria. For us to prevent huge numbers of getting infection, we mount actually prevention mechanisms like using mosquito nets or spraying so that you could minimize the concentration of the mosquitoes. Now, all those mechanisms of encounter will fail. And then the entry becomes very critical. Where somebody has a mic on, please switch off. Where is this pathogen going to enter? So the contact of a bacteria or a virus to the host becomes most important. And then there is issues of virulence functions. Many of them have got what we call virulence factors. Many pathogens have virulence factors, the factors which allows it to get into contact with the host and take a foothold within the host. They are called virulence factors. And some of them will have factors which allow them to adhere to the contact. These contacts are going to be either within the mucosal surface or the skin. Now, if it is within the mucosal surface, you understand that a number of them have fluid. It's gluing fluid. So this is able to clean away the bacteria and the viruses so that they don't stick. So that's a mechanism of ensuring you do not get infected. If it's skin, you need to understand that they will only enter on a damaged skin, and you have the epithelium, which prevent entry of the bacteria and viruses to the inside. And more often than not, the epithelium has a lot of dead tissue, which is actually shunned away. And when it's shunned away with multiples of pathogens. Now, in all that, a specific ligand receptor interaction, which allow the pathogen to interact with the cells of the choice, which will enable it to gain entry. Some of the virulence factors which you need to understand is like fibrillar, pili, surface protein, and all that. And obviously things like polysaccharides and lipotecoic acid, which are found in some of the bacteria which you know of. So these are important for you to understand. But the entry, we know the entry, and this is part of innate immune mechanisms. All those things you learned in innate immune mechanisms do prevent the entry of the pathogen. So skin, cilia, mucosal fluid, the pH becomes very critical in there for the prevention of the entry. Sorry, my cursor is behaving. Let me switch it off. Sorry, I'm having issues of moving my slides. So these are the areas of entry of most pathogens. And in each area, there are mechanisms which you learned in innate immune mechanisms, which will act as preventing the pathogens from entry. And they are all important for you to understand. So if you talk about the conjunctiva, if you talk the mouth, the GIT, the urinary system becomes very critical, as well as respiratory system and skin are so critical for them to understand. And each mechanisms in innate immune mechanisms, as you learned, becomes actually critical. Moving. So within all that, then you need, when you gain entry, the immune system requires to prevent the spread of that pathogen. It requires to actually prevent the spread of that pathogen if it gains the entry from the initial contact. Now, what are there to prevent the entry? We do have professional phagocytes. These will actually cause phagocytosis of the pathogen. We do have non-professional phagocytes, which will take up the bacteria through endocytosis and formation of phagolysosome. And two mechanisms. There are some bacteria which have gained mechanism to escape the phagosome, the hostile environment. There are some bacteria which have avoid that. They will escape the phagosome to the cytoplasm. And it's important to know some of those bacteria which will end up doing like that. Now, there are also bacteria which actually evade tissues. They can either degrade the extracellular matrix or they can disrupt the junctions. They are enabling them to move from cell to cell, simply to avoid the mechanisms which have been put in place against them. So the number one there, how the bacteria are going to evade is they can actually move from a phagosome to the cytoplasm. They can gain tissue to tissue or cell to cell movement, degrading the extracellular matrix or disrupting the junctions to enable them to move as quickly as possible from one position to the other. So those two will prevent the spread of the bacteria. Then if it happens to spread, then the next thing which will happen will be multiplication. The multiplication. Now, there has to be, for multiplication to occur, there are actually, you require nutritional environments. For example, in the lumen, in blood, urine, and within the intracellular as intracellular. So they require varieties of all these nutritional components. Now, the second one, which determines whether the bacteria is going to spread, will be the inoculum. How much did you get into contact with? The size of the inoculum. Now, the third one will be the incubation period. For bacteria or viruses with very long incubation period, sometimes it is not very good for that kind, for those kinds of bacteria or viruses, because the immune system will prepare. It is better if a pathogen has a short incubation period, because the immune system will not have adequate time to prepare to eliminate it. Now, a number of them will go on and acquire different elements, things like iron, transferrin, lactoferrin, so that they can be able to survive. And they will remove these from the host and to themselves, enabling them survival. Now, therefore, if you know all that, then the body mechanisms has been put in place so that it can evade, it can be able to destroy those pathogens. The first one is we teach you about within the mucosa is complement system. We know about that. Complement system is important. And it will be actually the opsonization. You remember C3b is very good in opsonizing. It will be the through MAC system, membrane attack complex. Now, if those mechanisms are there, then how do the bacteria survive? How do the bacteria and viruses survive within the complement system? They may actually simply not bind. They may not bind. So the antibody and antigen immune complexes may not be made for the complement pathway to be activated. They may actually not activate the complement system. Some of them, remember that like those ones, polysaccharide capsule, may actually not be able to activate the complement system. Then they may actually inhibit or deactivate the amplification cascade from C3 convertase to C5 convertase to MAC. They may actually inhibit that process. One factor which does that very well is factor H or M protein in streptococci. It does inactivate the amplification cascade of complement system. Now, it may keep activation away from the membrane. Lipopolysaccharides do that. They may degrade the complement system through secreting proteases, which will degrade the complement system. All those factors are enabling the pathogen to evade the immune system. These factors there are enabling the host to eliminate the pathogen. But for each function of the host, the bacteria have methods of evading those mechanisms. And it's important for you to know some of these mechanisms. And that's why I've gone stage by stage to document this for you. Now, but if the mechanisms, those mechanisms fail, if they fail, depending on whether the bacteria was extracellular or intracellular. Now, there are different mechanisms which are going to be put in place for extracellular pathogen, and this will either be, most of them are bacteria, a few viruses will pass through the extracellular pathway before going to intracellular. Then there is actually mechanisms they have put in place to survive. One of them is antiphagocytic functions. So a smart bacteria will inhibit recruitment of the phagocytic cells. They will inhibit recruitment of phagocytic cells. They will inhibit the complement system. They would inhibit the cytokines. All these are important in ensuring the phagocytic functions. Then they can kill the phagocytes. They can kill the phagocytes, and they can prevent the process of phagocytosis. They can prevent the process of phagocytosis. If it is intracellular, if it's intracellular, it's already inside the cell, the alternatives which it has is to inhibit fusion of phagosome and lysosome. When you inhibit that, you stay within the phagosome, but the lysosome will not come on board with its active enzymes, which destroys the bacteria. The second function it can do, it can escape the fusion just when the phagosome and the lysosome are going to fuse. That bacteria can escape to the cytoplasm. That it can inhibit oxidative burst, reduced oxygen radicals, which are produced within the phagosomes. And obviously, there are antimicrobial functions which are found within the entire phagocytes through phagocytosis. It can resist all those. It can resist all those. Now, if you want to understand how all those work, then you need to move from the process of phagocytosis. So within the bone marrow, smart bacteria will inhibit production of phagocytes. When the phagocytes are formed, it is important for them to fuse with the lysosomes to make phagolysosomes. Now, that process can also be inhibited. Now, more often than not, where there is damage, especially within the blood vessels, there is inflammation Now, those inflammation are either going to be caused by inflammatory cytokines, they can be deactivated, and therefore you end up inhibiting inflammation, meaning that now because inflammation enables the body to to ring fence the pathogen, meaning that pathogen can be able to spread if inflammation does not occur. Now, for the movement of things like neutrophils, macrophage, they can actually inhibit that movement, inhibit chemotaxis. It can kill the phagocytes itself. They can have mechanisms to kill the phagocytes or inhibit phagocytosis, inhibit lysosomal fusion, and resist killing. Those on this side are mechanisms bacteria and some viruses have actually very well developed within their system to prevent their destructions. Now, for you to understand this, then you need to go back and understand the process of phagocytosis right away from a movement of the pathogen, formation of pseudopodia, vacuole formation, phagosome formation, fusion of phagosome with rhizosome, and then digestion and residue formation and excretion. These 7 processes, each of them has a mechanism where some pathogens have evolved to prevent themselves being damaged using any of these mechanisms. So again, phagocytosis requires to be well understood. When I talk about the reduced oxygen radicals, this you have been taught in biochemistry about how it is produced within the cell. Now, please know that there are oxygen dependent mechanisms. One of them is pH, whereby actually you are going to produce actually hydroxyl radicals, which can fuse with chlorine to form acid. Then there is a lysosome, which are actually within the entire systems. They are proteins things like lactoferrin, B12, hydrolysis, which are all going to be produced within that system. This system, again, you need to remember it within the biochemistry, and most possible, you are still learning it if you are within all that. All that mechanism, which we have so far dealt with, revising about the mechanisms which have been put in place to eliminate pathogen, and the evolved mechanisms the pathogens have had to eliminate those mechanisms are important for you to understand about immunity. Now, the second one is now we go to the adaptive immune system, which technically is production of antibodies to have been put in place if anything is not handled by the innate immune mechanisms, is not handled about the complement pathways. Then you come to the antibodies and the cells. Then, if this is a hostile environment, the question would be, how then do the bacteria evolve around those systems? And there are a number of them when we talk about antibodies. There are a number of them when we talk about antibodies. One of them is what we call antigenic mimicry. Antigenic mimicry will services with the components which is antigenic mimicry. So when it covers its surface with the components which look like the host, it means the antibodies will actually mistake those components with the cell and will not end up eliminating that pathogen. Neisseria meningitis is one of the notorious pathogens for that. The second one is what we call antigenic cloaking. And this will simply actually enable the host proteins binding the surface of the bacteria. For example, protein A of Staphylococcus aureus. Then there is what we call, number 3 is what we call antigenic variation. You continuously change the antigens which are being generated. For example, Neisseria gonorrhea does change the surface antigens. Then you can have number 4, antigenic variety. So you can have a pathogen which produces numerous types of strains, and each strain is antigenically stable, which means the body has to recognize each of these pathogens, each of these antigens. And if you keep changing them, then you will end up exhausting the immune system and therefore gaining a foothold. The Streptococcus pyogenes is good at that. And you can easily actually make it very easy. Degrade the antibodies, just degrade the antibodies like Haemophilus influenza does. You degrade the antibodies, it will not be able to cause neutralization, opsonization, and all that. So these mechanisms, about 6 of them, are important for you to understand when it involves the antibodies, how the pathogen is actually going to evade those systems. The second mechanism is cell-mediated immunity. We know the cell-mediated immunity is the function of the cells, specifically T cells. Now, some pathogens can alter the host response. You can change whether it is going to be antibody mediated or a cell mediated. Mycobacterium leprae does that. And the 3 spectrums of leprosy, which I'm hoping you've learned from tuberculoid leprosy. The 3 spectrums are as a result of the pathogen changing the cell-mediated immune mechanisms evolved by the host. Now, the second one is damage. You can simply actually cause kill host cells. So a pathogen can decide to kill the host cells such that cytotoxicity does not occur. Now, toxins can do that. Or you can actually eject proteins which will cause apoptosis of the cell. Now, some of those toxins may alter the pharmacology or physiology of the host cells. And you can actually cause damage through influencing the host immune inflammatory responses. That can happen. And things like Streptococcus pyogenes does that very well, as well as rheumatic, you know about rheumatic fever, can actually change the host immune responses. I'm sure you know, if you have some people who have suffered from rheumatic fever, when the immune system is handling the streptococcal bacteria will end up actually causing the damage of the host tissues, That should not be very difficult. Now, some bacteria we know they actually form the toxins, endotoxins, like polysaccharide from gram-negative bacteria will actually is a toxin, and it causes damage of the host. And the cells which are going to be focused on are going to be actually the macrophage, which will end up producing tumor necrosis factor. We will end up producing interleukin-1 and interleukin-6. There are pathogens which produce endotoxins, proteins. And these can actually prevent different functions of the immune system. Some of them will actually alter the cells, like I did mention, killing the cells by altering their functions. Some of them will cause lysis of the cells. Some of them will affect CAMP levels. They will degrade the extracellular matrix of the host cells, or they can cause inappropriate activation of the T cells, and thereby the issue of superantigens. Or they can secrete type 3 secreted proteins, which when they are injected into the host cell will induce programmed cell death. And some of these proteins can also cause polymerization of actin, therefore changing the way different cells are going to be important. So it's interesting if you go and look around at a toxin, how they cause damage, how they actually inactivate the host cells, how they destroy the cells, as well as endotoxins, and then characterize some of the bacteria with either they are producing endotoxins or exotoxins. Now, but ultimately, a smart pathogen requires to move from one host to another host. And when it moves from one host to another host, it will have completed its survival mechanisms. But it is important for you to know that not all pathogens have that luxury of moving from a new host to a new host. There are some like Legionella pneumonia, which doesn't actually move from host to host. And it is also important for you to understand it is not the goal of the pathogens, smart pathogens, to kill the host, because then you are left without having anybody to infect. So some of the bacteria which cause disease have learned that balance of just being enabled to spread, to cause disease, and to infect pathogen, and to move from one person to the other. And those mechanisms they are going to use to move from one person to the other will be fecal oral route, sexually transmitted, droplet, skin to skin, fomite, or actually from mother to child. So again, these mechanisms requires to be actually known how a pathogen moves from one person to the other. So those are actually regarding the bacteria. I just have within the next couple of minutes remaining, I just need to paraphrase a few things which are specific for the viruses. First, it's important for you to know viruses are intracellular parasites. themselves, they can't grow independently like bacteria. They require host cell to multiply. And when they now get the host cell, they will be able to utilize the host cell to reproduce. They come in different sizes and shape and genetic component. And when you are taught virology, you hear about RNA, DNA, either single positive or double negative. So they do have a life cycle, and that life cycle will actually begin from infection and will pass through the extracellular component. So the way they are going to be handled when they are within the extracellular component is going to be the same way the bacteria are going to be handled. But many of them, the goal is to become intracellular. So the component of attachment, penetration, uncoating, transcription, replication, and assembly will actually require to be well understood when you are taught about that. So all that, for them to gain entry, again, they are going to meet the same things which have been put in place. The innate immune mechanisms they are going to encounter within the extracellular component, the innate immune mechanisms. So they have to avoid the fluid, the mucociliary excreta, the pH, the things like tears, urine, and all that. They have to avoid that, as well as actually epithelium. They have to avoid all that. And they'll only gain entry when they take a foothold of those innate immune mechanisms. Now, once they enter the body, the host, then there are actually antiviral mechanisms which have actually been put in place. Some of those antibodies, the same mechanisms of opsonization, neutralization are going to be critical. But then there is a concept of interferon. Interferon alpha and interferon beta, which are able to block the viral replications. And they are also actually able to activate natural killer cells, which do immune surveillance, and have antiviral effect. So natural killer cells becomes important in viral infections. Then there is phagocytosis, when during the gaining of the entry and infecting new cells, the macrophage will become the cells which are going to collect the dead tissue, and within all that, they will pick up the new pathogens and do the presentation to the T cells, the T cells. Now, when all that happens, then you need to understand that IgA is important. It can actually block the viral attachment within the epithelium. We do that with rotavirus, and which causes diarrhea in newborns. We do have other immunoglobulins, which may actually end up containing some of the viruses. And these functions are going to be having direct viral neutralizations. And these will actually abolish the infectivity of the viruses. And they can bind directly here. It can be through antibody-mediated complement lysis. This is actually going to be the classical pathway, antibody-mediated complement lysis. It can be through indirect mechanisms, through direct antibody interacting with viral antigens, the complement activation, opsonization, and antibody-directed cell cytotoxicity. Those mechanisms are going to be important in trying to ensure controlling the viral infections before they enter the cell, Once they enter the cell, the only mechanisms which is there to eliminate an infected cell is through destroying the infected cell, Why all those mechanisms we spoke before are not going to enter to the cell. Antibodies do not enter the cells. And therefore there has to be a different mechanism of killing the cells which have viral infection in them. And this is where now the CD8 positive T cells becomes critical in ensuring that they are activated through cytokines, being converted into cytotoxic T lymphocytes, which is able to target the infected cell and destroy them. The second cell is CD8 positive Th1 cells, which will secrete cytokines, which can actually act directly or indirectly against viruses. So the ones which cause inflammation will actually ensure that the area where the viral infection is surrounded. The ones which activate other cells will actually ensure that other cells are activated. Some of them will actually secrete interferon gamma, which has antiviral effect. Interleukin-2, which will indirectly act through recruiting new CD8 T cells, which will be converted into cytotoxic lymphocytes, targeting the infected cell, viral infected cells. The second cell, which is important in antiviral effect, is natural killer cells. They have antiviral effect. They can directly recognize the cells which are infected and destroy them. The third cell, which is important and is recruited, is actually macrophage. Again, macrophage can be activated and will be able to eliminate all the cells which have got intracellular infections. But through all that, the viruses have learned mechanisms of avoiding the infections. Most of them are just like we did in bacteria, but one of them, the specific ones, will include blocking protein synthesis. This is the host protein synthesis, so that it can be able to block synthesis of other proteins, and will utilize that mechanism only to make new proteins for new daughter cells. It can inhibit the presentation of peptides to CD8 positive T cells. So inhibiting MSC class 1 is important for viral infections. It can reduce the expression of MSC class 1 molecules within the infected cell, which simply means the cells are not going to indicate that they have an infection. It can block the activation of complement pathways, and it can change its antigen. And it can actually cause immunosuppression. HIV does that. Or it can suppress cytokine production. So these mechanisms are important for the viral infections, avoiding the immune system, all these. But the outcome of a viral infections will depend on the ability of the immune system to clear the viruses from the body. So it's a war, and the ability of the viruses to subvert this system. So the two of them will size one another. Whoever will win will actually end up becoming the master within that spectrum. So those are actually, you have 4 minutes, some key informations which you require. Class lab, are there questions?
I believe if there's anyone who has a question, they can unmute and ask you. If not, we'll just end.
Okay, I will send you this, but please remember, have you done microbiology?
Yes, we started with virology.
You started with virology. So somebody is asking about virulence factors, and I thought people in microbiology taught you virulence factors. Most pathogens have factors which enable them to be able to infect host cells. We call those virulence factors. Have you been taught about virulence factors, classmate? Yes or no? Class rep? Can anybody
Yes,
anybody
sir.
tell me whether you've been taught about
Yes,
virulence
we have.
factors?
Like, as we are doing the viruses, we normally like cover that as well.
Okay, very good. So I think there is no other question. That is the only one I could see within the chat. So can we say it, can we call it a day?
Yes, sir. Thank you.
Okay, you're welcome. Thank you.